Okay, students. So now we are going to today's lecture. We are going to talk about electrophoresis. So this is what we have learned so far. So we have learned the ion exchange chromatography and their utility in separating and purifying proteins of different charges. Then we have learned of size exclusion chromatography, where we have separated proteins using size. This can also be used for buffer exchange, size estimation, purity check. Okay. Then we have learned about affinity chromatography, where we have separated and purified proteins using affinity functional groups, and we have specifically looked at the example of histag proteins. Okay. The next one we will be talking about is 1D SDS page electrophoresis and 2D SDS page electrophoresis. So 1D SDS page electrophoresis, we already know the principle from last semester. It is used for analytical separations of proteins using size. So its utility is in size estimation and purity check. 2D is just page electrophoresis. So this is and we add another dimension. So we do analytical separation of proteins using both size and charge. So its utility lies in the fact that this provides an additional separation, and it's an excellent analytical technique. So SG spatial electrophoresis principle we already know from last semester, so I'm not going into detail in that. So how it experimentally works, you load proteins on well which are say linearized using SDS, so that have cumulatively negative charge on its backbone due to SDS binding to it, and then it is also got rid of any linkages like disulfide linkages by addition of reducing agents. Okay, so now if you turn on the positive charge, this entities will start traveling towards the positively charged due to its negative charge, and those that have more smaller positive charge, smaller structure, so they will travel fast through the gel due to because it travels through the pores, so this will travel fast, whereas bigger ones they will travel slow. So this will provide a size based differences. So something which is, for example, you can look at this gel, which is small molecular weight like lysozyme will travel fast, whereas mucin, which is large molecular weight, will travel slow. So what is the utility of this one? So this is a really good analytical, really good tool to find the purity of a protein. For example, this is a protein. So this can be tell that this protein is very pure. So purity estimation is one way, one utility of it. Second thing is it is used for size estimation. For example, if you run this unknown protein against a bunch of known protein markers, it can tell you that this roughly is what is the size of the protein. So we have done a lab on this, and we have quantitatively estimated using standard curve method that what is the size of this protein. Okay, so that we have done in the last semester. So size estimation is also what you can use this for. Great. Okay. So now what we want to know is that, so one shortcoming of SDSL page is that if you say have something like a cell culture run in the SDS page, this is what it looks like. You can see it looks pretty complex, right? Not like a really nice band. So you can see each of this band can have multiple proteins. Okay, so it is hard to differentiate them whether there are multiple bands present in each of these bands. Okay, multiple proteins present. So what you do, you use a method called 2D gel electrophoresis. So what is the principle of that? So this is what I was just suggesting, that the bands cannot be separated. The same bands, if they have same molecular weight, they cannot be separated, right? So for example, this band is a quite thick band. So how do you know there are not multiple bands there? So what we do here, we use a term called isoelectric focusing to separate them on an additional dimension, that is using the charge. So for this charge-based separation, what we use, you use their pH gradient and their individual isoelectric point. Okay. So here, for example, this is a 2D gel electrophoresis column. Okay. So here, what we provide something called a pH gradient. So this is done by something called an ampholyte solution. So ampholites are polyamines with different pK, and if you add them to it, they will provide a really nice pH gradient. So what's the pH gradient? It starts with pH 9, then followed by pH 8, 7, 6, 5, 4, and 3. Right? So that's a pH gradient. So you have to add this amphilites one at a time, and it provides a nice pH gradient. Okay, you can do this using also charge. So next, what you do, you add a protein solution to this. This is your protein solution being added. And then you start the electric field. So let's say you have a protein which is having an isoelectric field, isoelectric point of 4, that is your protein 1. Another protein which has an isoelectric point of 6, that is your protein 2. And another protein of isoelectric point, let's say 8, which is your protein 3. So remember what isoelectric point tells you, at this particular pH, this protein is neutral. So at pH 8, protein 3 is neutral. Right. So that means that once you mix this to the solution, so this protein 1 will start traveling. So all this will have the this will start traveling to the positively charged electrode, but it will stop its migration when it is roughly here. That is your protein 1, because once it is neutral, it cannot be moved towards the positively charged electrode anymore. Protein 2 would stop here because then it becomes neutral. Right? So protein 8 will stop here because then it becomes neutral. So it can no longer travel. Right, so regardless of what is the molecular size of these proteins, weight of this protein, so these proteins will first be separated using a pH gradient. Okay, so this does not really distinguish based on their size. So no size-based distinction at this point. Distinction at this point, or differentiation probably is the right word. Okay, so no differentiation at this point. So what you do then, so then you take this, so this is your outcome from the first run, that is your isoelectric point focusing. Then you put this on an SDS polyacrylamide gel. So this is your standard polyacrylamide gel. So now see, all these proteins are already separated. So let's say your this is your pH 9 and pH 3. So meaning if this is your pH 3 and pH 9, your protein 3 is somewhere here. Everything that is having pi 8 is somewhere here. Everything that has a pi of 6, that is here. Everything that has a pi of 4 is somewhere here, and so on. But this may or may not represent a singular protein. So this can be multiple proteins that would have that particular pi. Now, once you are starting the SDS page, now remember SDS page differentiates on the basis of size. So let's say there are 3 proteins here of different molecular weights. So molecular weights are 10,000, 20,000 and 30,000. Right. So once you separate them, you are separating them, the 10,000 here, then 20,000 here, then 30,000 here. Let's say here you had a protein of 10,000, you had a protein of 15,000, and you have a protein of 80,000. Right, so your 10,000 would be here, your 15,000 would be here, and your 80,000 would be here. But under normal circumstances, you would have not, on a normal SDS page, you would not been able to separate the 10,000 protein markers anymore. But under these circumstances, you are able to separate the 10,000 proteins from each other. Okay, so that is the advantage of the 2D SDS page. Okay, so this one here, for example, you can see that this is the actual SDS page gel. So you can see that it is the higher gradient and the proteins are being separated from this side to this side. Okay, so this is how it was done. Okay, so that is the decreasing molecular weight. So this is lower size. So this is, let's say, lower molecular weight, and this is higher molecular weight. Okay, so you can see. So if you have not done this, so everything that is, let's say, in this band would be forming one band. Everything that is here would form one band. Everything that is here would form one band, because otherwise they had different pHs, so they had been separate. But pI, isoelectric point, so that's why they are separate. Otherwise they would have formed a single band. So it's a really great analytical technique for separating proteins of different isoelectric point, and then with different size-based separation. So next we are going to learn, or at least starting to learn, something called a protein sequencing. So what is the protein sequencing purpose? So this is to characterize the primary sequence of the unknown protein. Okay. So proteins can be present written as either 3-letter codes, okay, or single letter codes. Okay. So this is your long protein. So let's say you have isolated this protein from a plant cell, okay, which is having a great medicinal value. So you have purified this protein using one of the previous methods, and now you are trying to find out what is the component of this protein. Okay, so what you do a sequencing. So first thing we would learn is the Sanger and Edman sequencing. So Sanger and Edman sequencing, these are useful to find the N-terminal amino acid or the amino terminal amino acid. Okay, so here what you have is a polypeptide, and you are treating this with this reagent called FDNB. So in the FDNB, this particular point, this particular fluorine is a replaceable fluorine, and this particular carbon is an electrophilic carbon. So what happens? This is your protein. So this protein's N-terminal, which is free, this NH2, this attacks this and replaces the fluorine. Okay, and you get this adduct. Then you hydrolyze with 6 molar HCl. Your entire N-terminal amino acid comes off in this adduct. Okay, so you can see here this is the side chain and this is the rest of the amino acid. So if you do a mass spec, you can find out what this amino acid is. Okay, so that helps to find out your N-terminal amino acid. An alternative way is using this reagent called phenyl isothiocyanate. Okay, so this is again an electrophilic moiety at this particular carbon. Okay, so because you can see it is attached with two electronegative atoms. So nitrogen quite and sulfur not so much. But the point here is that if you have an N-terminal NH2, this attacks on this carbon, and this makes this particular adduct. Okay, so next it forms this particular derivative or cyclic derivative of this terminal amino acids, and if you treat this with a CF3COOH, this relieves the rest of the protein. Okay, so this relieves the rest of the proteins. So originally, let's say it has the size of N. So this particular one would be N-1 because one of the terminal amino acid is forming an adduct. So similarly, if you do a mass spec, so the side chain is present. So if you do a mass spec, you can tell from the size that this is your adduct. Okay, so that is that. This is your N-terminal amino acid. Okay. So now we know that what the N-terminal amino acid, how to find out. So we need to get past one additional hiccup. So that is the presence of the disulfide bond. So our objective is to find out each and every amino acid in this. Okay, each and every amino acid. What are these present? Okay, each and every amino acid that is present in this long chain. So what usually cysteines do, they form disulfide bond. So that can complicate things because you would not know which cysteine is breaking down. So what we do, we break the next cysteines. Cysteines can be broken either by oxidation with the perfluoric acid, and it will form this sulfonate residues, or it can be reduced to diethioerythritol, which breaks down into SHSH and then can be acylation by iodoacetate. So that will form a this iodoacetate means ICH2 CO minus, so which has this electrophilic carbon. So this attacks here and replaces the iodine, and you get the CH2CO minus, CH2CO minus groups. Okay, the idea is to not let this go back to the disulfide linkage. Okay, so this should not happen. This should not happen. So we do something irreversible. Okay, so that's how you get rid of the cysteines. Okay, so now what you have done, let's say you have got you had a big chain, right? You had couple of cysteine linkages. Okay, so you want to sequence this, right? So you want to sequence this protein. You want to find out what each of these amino acids are. You had an N-terminal group. So what you did first, you find out what is the N-terminal amino acid, right? So you find out what is the N-terminal amino acid. So that we already know how to do it. Next, what you do, you treat this and broke down the cysteine linkages. Okay, so this is N-1 already. So you broke down the cysteine linkages. Okay, using the treatment that we just told you. Okay, so it can be SXSX I'm writing. So now what you have to do, you cannot really identify this. So what you can do, you can run a gel to identify its size, but that would not tell you what protein is present. Okay, so you have to identify it using by breaking it down into small peptides, then identifying those peptides. Okay, so breaking it down into small peptides and then identify this each of these peptides so that you can make the original construct. Okay, so I'll stop here, and from here we'll start the next class. Okay, and yeah, that's all.